Good morning, students. In the last class, we have discussed about proteins and peptides as drug delivery system. In that chapter, we have dealt what exactly proteins and peptides are and how they are very much useful in the process of treatment of disease. What are the proteins and peptides exactly we required for particular kind of a treatment of diseases. So, as we have discussed in the last class, what is mainly proteins and peptides are nothing but an what? It is an amino acids. It is a group of amino acids, and if the arranged in a particular chain system is called protein or peptides. Only the difference is peptides are just having this group of amino acid chain of less than 50. If it is exceeds more than that, then we will consider them as an proteins. It is in larger molecules, we can say in commonly. And we have studied what exactly the advantages and disadvantages we have with that, and what kind of treatment, like as I told, most of our hormones are nothing but then proteins, and the enzymes which are act as a catalytic for most of the reactions in our body, so especially for a metabolic process. So they are all are nothing but then proteins. So here we are considering what type of proteins and what kind of peptides are using particular kind of treatments. As we have discussed yesterday, what insulin is nothing but the protein. It is why it is required. It is for to reduce the or hypoglycemic condition, or it may useful for to decrease the glucose level. Hyperglycemic condition has to be reduced by insulin. So if there is a lack of insulin, leads to diabetes mellitus. So that is what the main thing is. So in the same way, like bradykinin is used for an treatment of labor pain, and some somatostatins are there for an ovulation process. So these are all are nothing but an hormones. So in generally they are all nothing but proteins and maybe in peptides. So what is the problems associated with the formulations of these proteins and peptides? Our intention here is, as a drug delivery system, we need to study what kind of protein and peptides we should take, and what kind of the drug delivery system we can form, so can we will deliver it in a proper way to for particular therapeutic action. The main thing associated with protein is we need to study about its stability, as how we have to study more stability aspect when it is comes about all other the dosage form. But specifically, there are two main things like emulsions. Emulsions are thermodynamically unstable dosage forms that you have already studied. So that is what the main thing is there. So the main emulsifying agents plays a lot of important role over there for to maintain its stability. So here also the stability aspect is more, instability is more with related to proteins and peptides. So to form them in a drug delivery system, we need to show what is their stability factors, what they are instability, and how we are overcome with that. So basically it will be classified into two types: physical stability issues and chemical stability issues. Physical instability, they may changes into denaturation process will takes place, adsorption will takes place, aggregation and precipitation. So here we'll see what exactly all these are one by one. Let us check exactly what they are. So denaturation is the process. We can see here the denaturation will takes place. Physical stability or instability will be, denaturation is nothing but what is a natural protein is the amino acid group associated with another amino acidic with a particular amine group bonding, or maybe a carboxylic group bonding. So that may be exists, so can it will form a long chain. So that is what natural. When during a certain conditions like variation in their pH, or may temperature, or may any other kind of organic solvent we have used, whatever the fluidity is there where the protein has exposed, with respective of that they may be changes their nature into another form. So that is nothing but called denaturation. For example, simple way in our natural process we can see when we have an egg, when we will make an egg, first it is initially what we have inside, a yellow yolk is there and then fluid is there. So albumin is most of the content. When we will be make it into an omelette form, how it will be? It will be a white, it is getting what we are getting, that omelette form is nothing but denaturation of that process. So similar way, that is what the thing is. That is the most difficult task. So we need to maintain that it should not be get denatured. So what is the thing we can see here? Any non-proteolytic modification. So modification will change us. So modification will take place, that is nothing but denaturation of the unique structure of a native protein that affects different changes in physical and also in chemical, and of course if they will change it into physical chemical, ultimately changes its biological properties. The main thing is this protein and peptides will compounds of polar amino acids and non-polar amino acid residues, both will be exist. Means they have an hydrophilic and hydrophobic part associated with them. So what are the factors will favors the denaturation is the mainly thing is one thing we can remain also based on that. If we will change, suppose the its nature is will be in a nature an aqueous state, when you changed into an organic solvent, there it will be denatured. So unfolding hydrophobic and hydrogen bonds are get broken, so in that way it may changes. And if the pH will varies, so what happens, is, so denaturation process will takes place. And even in the alteration in the ionic strength also takes place, and temperature rise is also matter for denaturation process. When you will apply an heat, there is a breakage of bond will takes place, that may leads to form of another structure, that may varies the structural act of the, that may leads to, that's what nothing but proteins, primary protein, secondary protein, they are nothing but a concept of denaturations only in generally. So denaturation may be reversible or it may be irreversible. Denaturation may lead to decrease in the solubility process, alteration in surface tension, loss of crystallizing ability, change in constituent group reactivity and molecular profile, vulnerability to enzymatic degradation, loss or alteration of antigenicity, and loss of specific biological activity will takes place. So that is what the major issues related with the denaturation. So how we can be prevent that? So we need to denature protein is we should restore on removal of an denaturants. Next, the main thing is physical instability is the adsorption process. The adsorption process, why we need to study here, the main thing is adsorption is nothing but what? The association of a molecule on one particular phase to another particular phase. So here it is an amphiphilic in nature. You know what exactly amphiphilic is, nothing but an the area at they will behave like an both the charges. Both the cases. So on that cases, there may be adsorption process, especially there is an interfacial phenomena. So suppose air water interface you can take, or the interface between the two immiscible liquids. On that case, it is having an capacity to miscible with the both the protein moiety. Suppose we have taken two immiscible organic solvent will be added in that, and at the interface it will behave like to be an natured with the associated with the one phase and also with the another phase. So that is what, because of that it will adsorb over their particular interface, that is called adsorption. So conformational rearrangement leading to denaturation can be induced by their interfacial adsorption. So denaturation ultimately, whether it is an adsorption or any other physical stability, alternatively what it will change, it will change its protein structural activity. So that is nothing but denaturation. At the end what happens is nothing but denaturation process. So after adsorption, they form some short-range bonds like van der Waals force, hydrophobic, electrostatic, hydrogen, ion bonds. So these are all the force of attraction between the molecules associated. So can the adsorption process will take place. You can see here that blue one will show the solvent molecule associating or adsorbing into an protein. It is called adsorption process. So adsorption of peptides and proteins at the interfaces are rapid, but the rate of conformational changes are little bit slower. On adsorption, there may be a loss or changes in biological activity as the molecular structure is rearranged. If peptides and protein drug entities are adsorbed at interfaces, there may be reduction in the concentration of drug available to elicit its function. That is what the major drawback. So on the basis of that adsorption phenomena, we must know at what extent this adsorption of proteins may take place. So such loss of proteinaceous drug may occur during purification, formulation, storage and all delivery process. So how can we prevent some of the examples we have see here? Insulin adsorption. Absorption if takes place, then precipitation will take place of insulin. That may be avoided by adding 1% or 0.1% albumin. So excess agitation should be prevented during production. The headspace within the confines of the container should be small. So some surfactant be used so can reduce the adsorption. Smooth glass wall best to reduce adsorption or precipitation. Next one is the aggregation and precipitation. Takes place with respective of adsorption or with respective of aggregation or with respective of precipitation. You can see here in the diagram as we have discussed, they have both the polar hydrophilic and non-polar hydrophobic tails. You can see this is the tail. In this sequence they will be associated. It is natural primary structure. This is hydrophobic. You can see the blue one, and it is an hydrophilic, the green one. So which will attract water. So when there is a solvent we are using, suppose we are using some hydrophilic solvent, then what happens? This all green colored moiety of the protein will be get associated with that. Then the formation will be changes, then denaturation. By this way, aggregations you can see here, the more one of the layer will be get aggregated with another. By this way they are again joining, and this is what leads to secondary protein, tertiary protein, that is all associated with their aggregation, or may they changes into precipitation. This is what the actual process will takes place that may leads to what instability of the protein. So if aggregation is on a macroscopic scale, it is nothing but precipitation. So it's only slight difference. Everything is same. Ultimately, they are getting aggregating. So if it is in macro scale, then it is precipitation. That is what, as we have discussed, this insulin will get be precipitated when they will be get adsorbed first, and it will go on. It will leads to aggregation. Then if it is go on further, leads to precipitation, like that. Interfacial adsorption may be followed by aggregation and precipitation, as I had told earlier. So extent to which aggregation and precipitation occurs is defined by the relative hydrophilicity of the surface in contact with the polypeptide. As I have told, what the solvent in which it is there, whether it is aqueous, hydrophilic content will be there, hydrophobic content or there. Based on that, the changes associated with the protein moieties will be takes place, leads to their precipitation and aggregation. So cause is nothing but what solvent we are using, what is the large air water interface generally accelerated,